and cell membranes. Part 1. Macromolecules and their functions. Macromolecules are the building blocks of life. They're made of repeating units that combine into large functional molecules. Carbohydrates provide energy and are found in cell walls of plants and bacteria. Lipids store energy and form flexible cell membranes. Proteins are made from DNA instructions and perform functions like enzyme activity and oxygen transport, e.g., hemoglobin. Nucleic acids, DNA, RNA, store and transmit genetic information across generations. Q&A, macromolecules. Q1. What are the 4 major macromolecules? A. Carbohydrates, lipids, proteins, nucleic acids. Q2. What role do proteins play in cells? A. They carry out many cellular tasks, such as catalyzing reactions and transporting oxygen. 3. Which macromolecule stores genetic information? Nucleic acids, DNA, and RNA. Part 2. Protein folding and denaturation. Proteins are chains of amino acids called polypeptides that fold into specific shapes to function correctly. Folding depends on hydrophilic, water-loving, and hydrophobic, water-fearing interactions. Denaturation occurs when proteins lose their structure due to heat, acid, or salt, making them non-functional. Cooking is a real-life example. Heat denatures proteins in food, making it safe to eat by killing microbes. Q&A. Protein folding. Q1. What is a polypeptide? A. A chain of amino acids that folds into a functional protein. Q2. What causes proteins to denature? Heat, acid, or salt. Q3. Why is denaturation important in cooking? A. It kills pathogens by unfolding microbial proteins. Part 3. Egg white example. Egg whites are mostly made of ovalbumin, a protein that turns from clear to white when heated. This change is from denaturation as the protein unfolds and becomes solid. This also demonstrates how heat can kill pathogens by disrupting protein structures. Q&A. Egg whites and protein. Q1. What protein is found in egg whites? A. Ovalbumin. Q2. Why do egg whites turn white when cooked? Because the proteins denature and solidify. Q3. What does this example show about proteins? That protein structure is sensitive to heat and essential for function. Part 4. Eukaryotic versus prokaryotic cells. Eukaryotic cells, larger, more complex, contain a nucleus and organelles, like mitochondria. Prokaryotic cells, smaller, simpler, no nucleus, no membrane-bound organelles. Both have a plasma membrane, DNA, and cytoplasm. Q&A. Cell types. Q1. What's the key difference between eukaryotic and prokaryotic cells? A. Eukaryotic cells have a nucleus. Prokaryotic cells do not. Q2. What structures do both cell types share? DNA, plasma membrane, and cytoplasm. Q3. Which type of cell is more complex? Eukaryotic. Bacterial cell walls. Bacteria are prokaryotic and have cell walls made of peptidoglycan for strength and shape. Gram-positive bacteria have a thick peptidoglycan wall. Gram-negative bacteria have a thin peptidoglycan layer and an outer membrane with endotoxins that cause more severe infections. Q&A. Gram-positive versus gram-negative. Q1. What is peptidoglycan? A. A sugar protein mesh in bacterial cell walls for rigidity. Q2. Which type of bacteria has an outer membrane? A. Gram-negative bacteria. Q3. Why are gram-negative bacteria more resistant to chemicals? A. Their outer membrane blocks antibiotics and enzymes. Part 6. Acid-fast bacteria. Mycobacteria. Mycobacteria, e.g. B and leprosy, have a waxy acid-fast cell wall made of mycolic acids. This wall makes them extremely resistant to antibiotics. Treatment takes months because drugs must penetrate the waxy barrier. Q&A. Acid-fast bacteria. Q1. What makes mycobacteria acid-fast? A. Mycolic acids in their cell walls. Q2. Why are they hard to treat? A. Their thick, waxy wall blocks antibiotics. Q3. Name a disease caused by mycobacteria. Tuberculosis or leprosy. Part 7. Cell membrane structure. The plasma membrane is a phospholipid bilayer. Heads face water, hydrophilic. Tails avoid it, hydrophobic. Proteins in the membrane help transport substances in and out. In bacteria, this membrane is fragile and needs the cell wall for protection. Q&A. Cell membrane. Q1. What is the plasma membrane made of? A. Phospholipids and proteins. Q2. Why is the cell wall important for bacteria? It protects the delicate membrane from bursting due to water pressure. Part 8. Lysozyme and antibiotics. Lysozyme is found in tears, saliva, and egg whites. It breaks down peptidoglycan, especially in gram-positive bacteria. Penicillin also targets peptidoglycan, weakening the wall so the cell bursts. Q&A, lysozyme, Q1. Where is lysozyme found in the body? A. Tears, saliva, egg whites. Q2. What does it destroy? Peptidoglycan in bacterial cell walls. Q3. Which bacteria are more susceptible to lysozyme? A. Gram-positive bacteria. Part 9. Bacterial shapes and arrangements. Shapes: cocci, spherical; bacilli, rods; vibrio, curved; spirilla, spirals. Arrangements, mostly in cocci: diplococci, pairs; streptococci, chains; staphylococci, clusters. Q&A, shapes and arrangements. Q1. What shape is a vibrio? A. Curved rod. Q2. What is the arrangement of streptococci? Chains of cocci. Q3. What does staphylococci look like? Grape-like clusters. Part 10. Flagella and motility. Flagella help bacteria swim. They spin like propellers, allowing bacteria to run and tumble. Not all bacteria have flagella. Those that do are motile. Q&A, flagella, Q1. What structure allows bacteria to move? Flagella. Q. How do flagella move? A. By spinning like a propeller. Q3. What is tumbling in bacteria? A random change in direction to reorient movement. Part 11. Endospores and bioterrorism. Endospores are survival structures made by Bacillus and Clostridium, resistant to heat, bleach, and radiation, remain dormant until conditions improve. Anthrax spores are an example, can be used in bioterrorism due to their resilience. Q&A. Endospore 1. What triggers endospore formation? A. Harsh conditions like lack of nutrients or water. Q2. Why are endospores dangerous in bioterrorism? They are extremely hard to kill and easy to spread. 3. Name a bacteria that makes endospores. Bacillus or Clostridium. Part 12. Final comparison, prokaryotes versus eukaryotes. Both have DNA, cytoplasm, and membranes. Prokaryotes, small, no nucleus. Eukaryotes, larger, complex, have organelles and nucleus. Hash Q&A, cell comparison recap. Q1, what's the main structural difference? Prokaryotes lack a nucleus. Eukaryotes have one. Q2, which cell type includes human cells? Eukaryotic. 3, do both have membranes? Yes.